for the good words said about me. Yes, I'm proud to be associated with the YCAS Satara. Thanks for your compliment. Thanks for your wishes. Without your blessings and the wishes, it was just not possible for me to reach the designation where I am today. Basically, my roots are from the Science College Satara. As the roots have been nurtured very nicely, it can bloom in coming times very nicely. This is a beautiful example. The way my teachers have taken care of me. Professor Viva Deshpande sir was my beautiful teacher during my graduation. We had a number of, we can say, trips also there. Again, during the research activities, we had been to a number of places in the participation in the conferences, symposia, so many things are there. Even we do have a number of research papers published together. So without the help of such beautiful teachers, it was not possible for me to reach that particular designation. So I take this opportunity to acknowledge Professor Deshpande sir, now in the capacity of the Vice Principal, and all the staff of the Department of Zoology, YCIS College Satara, for giving me a beautiful opportunity to groom in my life. Thank you one and all. Now with this, coming to the today's presentation, I would like to first of all acknowledge one and all for starting the MSc in Aquaculture and Fisheries from this year onwards. So the faculty development program for the syllabi related to the LSFT 101 Biosystematics and Biodiversity. It is carrying the credit, theory 4, practical 4, and total theory lectures are 60. I hope the slide is going ahead.
No, the slide, it's not moving.
Okay, please hold on, sir.
Yeah, it's okay.
It's okay?
Yeah, okay.
Okay, sir. Okay, now when we say biosystematics and biodiversity, you all are teachers, so I did not go in the minute details of all these things. You are having number of experiences, I can say the decades of teaching experiences, and then we are just coming as a part of faculty development program. So I will just go on the overall what one has to teach or what the students are expecting from this particular paper. I will be emphasizing on this particular aspect only. So coming to the very first slide, when we say the biosystematics is a combination of number of things. And if you can see all these things, we have the biodiversity, we have the management conservation, medicine and pharmacology, biochemistry, genetics, biotechnology, politics is there, survey and monitor is there, evolution, biogeography is there, agriculture, forestry, fishery, ecology, environment, all these components which are present in the ecosystem leads to the biosystematic. The word itself indicates bio means life, systematics means to arrange them in a very systematic manner so that one can understand what is there. That is biosystematics. Now, if you look, the biosystematics are in the vernacular language, or we can say for the common man. Common man is not using the word biosystematics. Generally, we use the word taxonomy, or we can say broader aspects, classification. For example, classification, vertebrate, invertebrate, number of things are there. Invertebrate, again, number of things are there. But when we come up for the MSc students, or we can say when we come up for the postgraduate student, let's have the better language than using the taxonomy. Instead of using the taxonomy, if we use the word biosystematic, that is a graceful way of taking the student to a bit higher level than the graduation. That is what I feel the teacher should go for, using the synonymous words as far as the concept, but the synonymous word is of little bit of higher version than what we are using the words during the graduation, because now the students are coming for the post graduation, and if the teachers are elevated, obviously we can take the student to a higher level than what they are for the graduation level. So this is just an example of what I am saying. Instead of using the term taxonomy or classification, if you go for using the biosystematics, initially it will be a bit difficult for the student to digest, but in the course of our repetitive we can the pronunciation of the biosystematics, they will understand, and then the word biosystematics will become part of their everyday language. That is how we have to groom the students to the above level, or we can say bit higher level, I can say. Now, if you look at the tree of the life, right from the soils, everything started, viruses, bacteria, archaea, eukarya, so many things are there. And finally, we have the common stem from which we are branching. From here up to the mammals, we can go. And what I'm saying is that when we say tree of the life, yes, we originate at the end of our life, we get mixed up in the soil. So if we, in this way, we keep on giving the example to the students of the day-to-day life. If we mingle the society, if we mingle the day-to-day activities with our teaching, then the subject will be very easy for the student to digest and assimilate. Now, for example, if I say here, okay, the life originated from the soil. And what happens to the individual? For example, mammals on the top of us, we as an individual, human beings, Homo sapien as an individual. What happens to the individual after the death? Again, they mix up in the soil. So this is how we can make a complete circle of the life to the student, and then the student will slowly and gradually get the grip on the subject, and he will start liking the subject. The most important thing in the biosystematics, or we can say for that matter, any subject for the MSc, we should create the interest in the student. If we make the student thirsty for the knowledge, and then he will go for the where is the water, where is the water for the knowledge, where is the water for the zoology or biosystematics, any paper for that matter, we need to create the urge in the students. That is what is expected from the teachers for the students. Now, if you go for the levels of the classification, generally we don't use the word domain. We start our classification from the kingdom, phylum, class, order, family, genus, and species. And yes, most of the time we can say yes. For the MCQ, we use the word which of the following is the lowest rank in the levels of the classification. We put so many things, and species is the correct answer. Or the vice versa, when we are teaching the student, we should also feed them the MCQ, which are the possibility of coming in the upcoming examination. We all know that we are going through the pandemic, so it is. I don't think so. It is possible for us to have the subjective type of examination. So obviously, we have to depend on the MCQ. While our teaching, if we keep on saying, now for example, which of the following is the highest rank in the levels of classification? The answer is kingdom. So in this way, in our teaching, if we keep on giving the examples of the MCQ, that will get attached to their brain from time to time, instead of giving them a big question bank. This is the 100 MCQs, of which some 20-25 are going to come for the examination. If we teachers have this type of attitude, then what happens? We are making the concentric circles in the teaching of the student. What the student will do, he will either mug up all the 100 MCQs or memorize the answers of the 100 MCQ. And whenever the question paper comes, he will be just what we can say, reflecting the answers of what he has mug up. And in the short memory, immediately after the examination, the student will forget. So our job as a teacher is not to make them mug up or not to limit their way of thinking. We need to make them think on their own. So instead of giving them MCQ from time to time, or instead of giving them the question bank for the upcoming examination, let's teach them the MCQs or what are the possibilities of the examination along with our teaching. So we are not saying that this is a question bank. We are just giving them a number of MCQs, and at the end of the term or at the end of the semester, few of the MCQs which are present will be given to them. Because if we give them the question bank, we are, I think, limiting them. We are not allowing them to explore what the actual subject is. This is the humble request to all the faculty members involved in teaching the MSc. Now, if we look at the taxonomic characters, we can involve the students like we have the morphological characters, anatomical, embryological, paleontological, molecular, biochemical, and ecological, geographical characters are there. And then we can give the example. If you look at the morphological characters, what are the similarities and differences? If you look at the anatomical characters, we do have the, for example, synonymous organs. For example, we have the hands are there, in the birds, wings are there, and in the aquatic mammals, the flippers are there. From the time to time, if we generate the interest in these terminologies, initially when the student come for the MSc, they're scared of such things. For example, morphology, anatomy is quite good, but when we say paleontology, they might not have heard the word paleontology. So we need to create the interest so that in future, these MSc students are the raw material for the research, or we can say these are the raw material for the PhD stakeholders in the coming times. So we are grooming the student so that they can come for the research, so that they can be the good faculty members in the society. For that, we need to make their roots very strong. Let's not take care of the what we can say flourishing flowers. We need to take care of the roots. If we make the fruits of the students very strong, you don't have to bother about the fruits and the flowers in the coming time. The student is bound to grow in his life if the roots of the education are very strong. So for that reason, we need to what can inculcate the concept of understanding the basic terminology from time to time. So as far as your syllabus is concerned, the unit number 1 deals with the taxonomy. I will not go into much detail. Introduction to taxonomy, stages and importance of taxonomy, problems, aim, and tasks of the taxonomy. So many things are there. So there are 4 different bits in the unit number 1 taxonomy. Now, when we say morphology, instead of students, they understand the pictures more. If we start the morphology, we say just the term morphology. Instead of that, if we depict some pictures, we can say instead of saying 100 words, 1 or 2 pictures speak more than the words. So let's explain them in the concept of the pictures more. For example, here I have given some example morphology. Similarities and differences in the shape. If I show the earlier slide, what the morphology is. Now, if I show them the pictures,
Excuse me, sir.
how? Yes, sir, please.
Hello.
Yes, हाँ बोला बोला की
Hello, sir.
बोला ना प्लीज.
Slide is not moving, sir. So,
आता झाली का?
no,
आर यू
sir.
एबल टू? नाही झाली.
नाही झाली. Okay, sir. Yes.
आता मॉर्फोलॉजिकल साईड दिसते का?
Yes, sir.
आता पुढची अॅनाटॉमिकल दिसते का?
Morphological दिसते आता, sir.
आता अॅनाटॉमिकल
आली.
आता दिसते का? शॅल आय गो इन दिस वे देन? आता अॅनाटॉमिकल
Yes,
दिसते का
sir.
सर?
Yes, sir.
ओके, ठीक आहे. आता मी अशाच पद्धतीने ठेवतो, कदाचित झूम केल्यामुळे ते
Yes, sir.
पुढे जात नसेल. ओके, लेट्स गो इन दिस वे डझंट मॅटर. Now let's come to the another aspect of the anatomical. Anatomical means simulate the differences we can see. If I move my cursor over there, we can see the human is there, dog is there, birds are there, some whales are there, and we give them the synonymous names. For example, this is the forearm, how it is developed in the dog, how it is in the terms of the wings, it is developed in the birds, and when we speak of the mammals, it is in the terms of the flippers. So that is how we can explain them more with the pictures. Now, embryological similarities. If you depict this picture to the students, students will be very happy to see. See similarities during the embryonic development. So this is how I look like at the time of my birth. You have to tell them कि in your life, do you remember the first picture what you can recollect? तुमचा सर्वात पहिला पिक्चर तुम्हाला किंवा फोटो तुम्हाला तुमच्या आयुष्यातला आठवतो का? This is how we have to carry the sweetness of the life along with our teaching. If we join the day-to-day activities, if we join them realistic, what you are enjoying in the life, if we depict them in our teaching, I think that will be more wonderful, that will be more graceful while teaching the subject in a very monotonous way, or we can say just for the sake of the teaching. Just yet come for the MSc, one of the paper is the biosystematics and biodiversity. In not that way, tell them to remind, do you remember your picture? For example, if I say, oh, I remember the picture of my class at the first standard. I remember when the naming, what we can say, नाव ठेवणं बारसं ज्यावेळेला होतं त्यावेळेला माझा फोटो माझ्या घरामध्ये आहे. मी कसा त्यावेळेला दिसत होतो. You know, in terms of vernacular language, in terms of English, in terms of all these things, if we carry the student with us, always the student will love to come to the class, and he will not dodge the class, or we can say he will not come to the class just for the sake of the attendance, but he will have urge to learn number of things. Now, for example, embryology. If we ask the students, suppose the 20 or 40 students are sitting in front of you, please tell me who can remember the first photograph. जसं मी तुम्हाला आता सांगितलं की माझं पाळण्यात नाव ठेवताना माझा फोटो माझ्याकडे आहे, किंवा त्याच्या आधीचा कुठं फोटो आहे का तुझा. So many things are there we can relate with our teaching. Now, this is the molecular similarities are there. Coming to the biochemical, different organisms have the different chemical composition. Here we can give the example. There are the fruits. Here I cannot give the examples of the difference between the flesh of a fish and the flesh of the prawns. This, to certain extent, I avoided giving that example. Here I have taken up the example of the mango and the tamarind. See, both are the fruits, but if we taste the mango, it is very, very sweet. And tamarind, the moment I say the word since, असा जर शब्द मी वर्गामध्ये उच्चारला की नाही, सगळ्या विद्यार्थ्यांच्या तोंडाला पाणी सुटतं. कारण प्रत्येकाला माहित असतं की आपण लहानपणी दगड मारून चिंच काढलेली असते, आणि चिंच ही आंबट असते. टॅमॅरिन हा शब्द वापरला तर कदाचित त्यांच्या तोंडाला पाणी सुटणार नाहीये, पण मी चिंच हा शब्द जर वापरला आणि तेही टिपिकल टोन मध्ये जर वापरला ना, ऑब्व्हियसली सगळ्या विद्यार्थ्यांच्या तोंडाला एक पाणी सुटतं. अँड दे हॅव अर्ज, यस, दे रिमेंबर देअर चाइल्डहुड, की आपण कसं एखाद्या झाडाच्या इथं दगड मारून चिंच तोडली होती, so many activities are there, and then we can come to the biochemical composition. Why the tamarind is of that particular taste, why the mango is sweet in taste. In this way we can take the student along with us to complete our syllabus. Ecological, geographical locations, why there is the thick forest is there, on the other hand why the deserts are there, what type of water is there. For example, we say water, drinking water, generally students are using the water, okay, drinking water, it is actually not a drinking water, it is a potable water. Now, if I say marine water, obviously all the students will say it contains the salinity, brackish water. It also contains the salt. But if I ask them one question, does the fresh water has the quantity of salt in that? Most of the students will say no. The answer is not correct. It has the salt in terms of 0.5 ppt is called as the fresh water, and the fresh water is again divided into the potable water, and so many things are there. In that way we can take the students along with our teaching. And if you look at the number of species known to us, as far as the zoology is concerned, maximum insects are known to the human beings, and the minors are just the protists which are known to us, or we can say the single-celled organisms are known, because most of the work in the taxonomy, most of the work in the biosystematics is done for the insects. And what is the study of insect called as? It is called as the entomology. So this is how we have to relate the higher version of the education to the students of the MSc, so that they will understand the things in a better manner. Now, for example, if you come at the what is the ecosystem, what is the biosphere? Biosphere, bio means life, sphere means circle. This is how the organism is. Single individual, we call it as an organism, and the group of organisms coming together is called as a population, and different individuals coming together, this is called as the community. And the community, when it interacts with the biotic and abiotic component, it is called as the ecosystem. And where is the ecosystem present? Ecosystem is present in the biosphere. Sphere because it is circle, bio means life. This particular circle is full of life, so it is called as the biosphere. This is how we can take the subject to the students. Again, this is called as the realm of ecology. If you look at this particular bit, it is called as subatomic particles. But it is the earlier terminology of the subatomic particles. Now we are making use of the word called as the nanoparticle. If I use the word nanoparticle in the class, and if I ask what is nanoparticle, 10 to the power of what? 10 to the power of minus 13 is called as the nanoparticle. So this is how from time to time we can tell. And in the MCQ also, which of the following is called as the nanoparticle? 10 to the power of this much, 10 to the power of this much, and one of the answer is very correct. So instead of using the subatomic particle, we can tell them the nanoparticle. So this is how from the nanoparticle up to the universe we have the things. Now, as far as the zoology is concerned, we are interested only in this particle, called the organisms, populations, communities, ecosystem, and all these things are present in the biosphere. But you see the thirst of the man. When we say the biosphere, biosphere means what? The life is present in the particular sphere. So it is called the biosphere. But now we are going beyond the earth. We are going in search of the life. In the terminology we say in search of the alien life, sometimes we say. Alien life, whether it is present beyond the earth, whether it is present on the Mars, whether it is present in the solar system, or beyond the solar system, what is there? Beyond the solar system is the galaxy. Beyond the galaxy, we have the universe. Then again, the question comes, are we alone in the universe? Now, if this particular question is asked to the students, give them opportunity to find the answer. Are we alone in the world? Are we alone in the universe? You must have seen the number of movies which deals with extraterrestrial life. ET, we call it as a extraterrestrial life. Aliens are there. So many good Hollywood movies are there. You recite them few of the stories of the then you come to the Discovery Channel again at some 9:30 or 10 p.m. We do have some episodes called as the Contact, Aerial Life, Wonders of the World, Secret of the Science, uh, World Beyond, World Below the Earth. So many episodes are there which are related with the National Geography, which are related with the Sony BBC, which are related with the Discovery Science. This is how we need to tell the students, look beyond the horizon, look beyond the vernacular way of listening to the music. It's very good to listen to the music, but now when you come for the higher education, look at the National Geography, look at the Discovery Science, look at the Sony BBC, all these channels you are supposed to see on the television. And again, gracefully tell them, instead of going in the Hindi language, all the, I think, we can say soft TVs, or we can say higher version of the TVs, they are having the language modification. Watching all these programs in the Hindi or watching these programs in the Marathi is not a graceful thing. If you want to learn the subject, listen to them in the English language. And now see the difference between when they're narrating in the Hindi and when they're narrating in the English. You will see a great difference. When the same concept is going on in the Hindi, more of words are there, and you are focusing on the words rather than on the what is going on the TV. Come to the English version. You can see few words are narrated and your full attention is there on what is there on the screen of the TV. This is how we need to groom the student from time to time to understand, to increase the horizon of their thinking. That is what is expected from the teachers for the students who are upcoming for the MSc. Now the learning outcome, everything is there with you. Student will be able to define the taxonomy and the various tasks of the taxonomy, so on and so forth. Coming to the unit number 2, the concept of species. Again, the syllabus is available on your desktop. I think the syllabus is also available on the website of the YCIS Satara, so one can go through it. I will not go into the minor details of the syllabus. Please hold on, something has gone wrong at my end. Yes. When we tell them what is mimicry, the resemblance of an animal species to another species or to the natural object, for example, the praying mantis. Now, if you go in the field, we can see this will look like a stick, but for the MSc student, we can see it is not a stick, it is a praying mantis which has a mimicry of a, we can say, a dried stick so that it can survive. Another beautiful example of a chameleon. For a common man, it looks like as if a dried leaves, folding of the dried leaves is there. But for the zoologist, we can say it is a chameleon. Again, we can say a horn owl is there. So many things are there which we can teach the children from time to time. Coming to the unit number 3, biodiversity science, evolution of biodiversity. And the second part is the evaluation of properties for the conservation of habitat and the species, so on and so forth. Now, this is how the common insects damaging the crops are there. So many insects are there which are damaging the crop, but why they are damaging? Are they really damaging? They are not damaging. It is survival of the fittest. Insect also want to survive, human beings also want to survive. Now there is a conflict between the survival of the insect and the survival of the human being, and we as a human being label some of the insect as pest, while other insects we label them as a useful insects. For example, honeybee is there. For example, number of birds are there which are pollinating the things. For the welfare of the human beings, we are segregating as them useful insects, harmful insects, or pests, and we make use of n number of chemicals to control them. But please remember, they also want to survive. As we want to survive, they also want to survive, and that is the reason there is a conflict. And obviously, man, he wants to survive better than insects. He's least bothered about the biodiversity in the world, so his dominance is more there on the planet Earth. This is how we can correlate by saying so many things like pest is there. Basically, pest is not a pest. They are living their own life. We are living our own life, but we, as they are harmful, we have labeled them as a pest. Basically, they are not pest, but they are just insects which are feeding on the crops which we need to cultivate for ourselves. So we can see the impact of borers in the forest. We say trees are falling down, felling down. We can say it is because of the natural calamity, like the borers are there. Damage, extensive damage. We can say now we don't have to fly in the air, but the concept of the drones is there. When I'm using the word drone in the class, again, the horizon of thinking, the way the students are looking at a particular thing, their vision is expanded. They come to know something is there called as a drone. With the help of drones, we can have the aerial view of everything. Even if I go in the satellite picture, if I type the satellite image of YCIS Satara, You can have the satellite image of your own college. So this is how we need to inculcate the way of understanding the concept to the student. And then whenever the students are free, I think now all the way everybody is having the smart mobile or a smart handset in them, and you can make use of the technology, not just for the personal gain, but to understand. To increase the horizon of the thinking, we can make use of that technology. So when I say the word drone, they understand, oh, it is something. With the help of drone, can we have the picture of the YCS College Satara? Yes, you can. So this is how we need to explain the concept from time to time. Again, one insect feeding on the another insect. Survival of the fittest is there. But as far as the human being is concerned, we label it as the biological control of the pest. In the taxonomy, we say biological control of the pest. But in the nature, what is there? One insect is feeding on the another. It is the food of that particular insect. So it is a survival. But from our point of view, biological control of the pest. The most significant and the least harmful as compared to the chemical is the biological control. That is how we inculcate the subject to the student. Again, another example is there. Learning outcome is there. That comes the last unit, that is the biodiversity conservation is there. NGO, when we say role of the NGOs, it is a non-governmental organization. What is the interaction of the colleges and the universities along with number of NGOs are there, even at the Satara also. Satara is placed very close to the, we can say the Caste Plateau is there. And we can tell them why Caste Plateau is very famous because of the biodiversity which is present over there. Now saving the world's food seeds, the global seed board. Here we can give an example to the students. We can say, now for example, most of the faculty members, those who are listening to me, are in the somewhere between the 45, 50, this age group, or 55, this particular age group, we all faculty members are there. For us, the songs of the Kishore Kumar, the songs of the Asha Bhosle, the songs of the Lata Mangeshkar are the melody for us. We often feel to listen to them when we are going for the morning walk or when we are at the leisure. But for the generation which is sitting in front of us, for them, remix is the original song. Why the remix is the original song for the students? Because they have never ever heard the original song. The same thing I can correlate it with the food which we are eating. The original food which is there, for example, the original basmati rice, or let's, if I give the example of the Kolhapur, I can say the Gansal rice of Kolhapur, or Ratnagiri Chuvis, the rice of the Ratnagiri. They are the original breeds of the rice, but due to the hybridization, due to the climatic conditions, the seeds are getting deteriorated, and we are feeding just proteins, lipid, glycogen rather than having the nutritious value. Now, where is the original seed present in the world? Give this example. Here is in the Norway. The vault is there deep in the cold region. This is how there all the original seeds of the world, including that of the Kansal rice and Ratnagiri chuvis, all these seeds are available. And yes, if there is a natural calamity, we can get these seeds and grow and regrow so that we can fulfill, or we can say feed the entire world. But if we don't know what the originality is, we just keep on, earlier example I have given, remix songs are original songs for the upcoming generation or the generation which is sitting in our classroom. They need to listen to the original songs. They may not like it because now their choice is different, and we cannot ladden our choice on these new generations. But they must be exposed to what the original concept is there. Here I can say regarding the biodiversity also. Learning outcome, so many outcomes are there. And yes, when I have gone through the syllabus, I have seen number of recommended books are there. But for all the teachers, I will humbly request you all. There is one beautiful book called The Theory and Practice of the Animal Taxonomy by V.C. Kapoor, the finest book in biosystematics, and I think entire syllabus is also based on the book of the VC Kapoor. No doubt you can have the, you can go for the number of books are there, even the internet, Wikipedia, so many sites are there. But this particular book, I will recommend it, the authorities of the YCIS Satara to maintain the hard copy of this particular book in your library, and see that these books are also made available to the student in the either in terms of the soft copy or xerox of the hard copy also. But this is the best book if you want to understand the biosystematics and biodiversity, because I am involved in teaching this particular paper for last 5 years. So I am using this particular book, and I always prefer it. It has a lucid language, it has a very simple language, and things are simplified in a very simple manner. So thank you very much for the first part, that is how we have to deal with the theory part while teaching to the students. Now I will come to the practical part. Now coming to the practical based on the LSFT 101. If you go for the syllabus, I can say the study of museum specimen is there from vertebrate, invertebrate, one representative from each class, so on and so forth. This is the list of the practicals which has been taken up for the syllabus of the MSc Part 1 Aquaculture Fisheries Specialization. I will not go in the detail, but yes, so few of the examples I can share with you. For example, study of museum specimen from the invertebrate phyla. As far as biosystematics and biodiversity is concerned, this is how we have to show them the picture. And yes, in the pandemic, it is not practically possible to bring the student to the laboratory, but this is how we can show the picture. This is sketch and label in your journal. But how it looks in the nature, the way the individual looks in the nature, this is how the individual looks in the nature. You must know how it looks in the nature and what the sketch and label is there in your journal. Two different things we have to make them very clear. First, sketch and label, and second, how it looks in the nature. Another example of euplectella is there. This is sketch and label, and then on the other page you have to write the description. And again, give them instructions. Never use the pen on the drawing page. I have seen number of students, no doubt they are drawing with the pencil, but when it's time to label them, they are making use of the pen. It is very horrible to see the students of, even at my end, the MSc Zoology student, they are labeling with the pen. Basically, the drawing page is meant for pencil only. So make use of the pencil even for the writing. These are the small instructions which we need to give to the student from time to time while conducting the practicals. And this is how the actual animal looks like in the water. Deep water, this is how it looks like. And the beautiful story goes with it. This is the prawns with the labeling, and this is how the exact prawn looks like. Another examples of the vertebrate phylum is there. This is how the electric ray is there, sketch and label, but in the nature, it looks like this. Flying fish is there, in nature, it looks like this. So we need to correlate what we are drawing on the journal and what is there in the nature. We need to correlate them. If they're coming to the department, well and good. We do have the museum specimens preserved either in the stuffing or in the form of the formalin. We do have the number of specimens in the museum, or we can say at our laboratory. But if we have to go them on the online teaching, this is where we need to tackle the scenario today. Today's pandemic can be handled in this manner. For example, bat is there. Some of the endangered species are listed, for example, snow leopard. One can give the description, one can give the actual photograph, and this is what you have to sketch and label in your journal. Coming to the adaptive modifications of the beak in the bird, give the description of what the seed-eating beak is, show them the picture of the sparrow, look at the difference between the beak. Here, I'm not focusing on the entire sparrow. The concept is focus on the beak. That is how we have to tell the students. And another examples of the beak modification, cutting beak is there. Look at the example. Now comes the difference between the jungle crow and the domestic crow. In this way we can, if let's focus on the beak of, see the pointed beak is there as far as the jungle crow is there. But if we compare this particular beak with the domestic crow, again you can understand the difference between the two. Adaptive modifications in the feet, cursorial or running feet is there. We have the examples of the emu is there. Modifications in the animals, blood fluke is there. This is how the blood fluke looks like. This is how the life cycle of the blood fluke takes place. And when we look at the symptoms of the blood fluke in any patient, these are the symptoms of the blood fluke. But when it comes to putting in the journal, this is the picture which you have to put in the journal. So what I have done, I have exhibited the original slide of the blood fluke. I have given the life cycle of the blood fluke. I have given the symptoms of the patient suffering from the blood fluke, and the actual diagram the student is supposed to draw. Again, we compare adaptive modification in the mouth parts of the insect. See, here we can see the mosquito anopheles female is there. बराच वेळा we can say की सातच्या आत घरात खूप छान पिक्चर असतो. अलीकडच्या मुली सातच्या आत घरात येत नाहीयेत, परंतु फिमेल मॉस्किटोज आपल्या घरामध्ये लवकर येतात. सी again the student will be very attentive. 7च्या आत घरात मुली येत नाहीत परंतु डास आपल्या घरामध्ये येतात. In the vernacular language, if you day-to-day examples, if you take them, definitely I can assure you the student will have urge to understand. He will lifelong, the student will remember that they are the female mosquitoes which are coming in the home to bite us. The male mosquitoes are not coming in our homes. Male mosquitoes feed on the plant sap. Female mosquitoes, they come in the home. So when we give them the examples like this, Sachat Gharat, lifelong they will remember. What they will remember? The mosquitoes which are biting in our home are the female mosquitoes. The female species is feeding on the blood of the human being, while the male species are feeding on the sap of the plants. If I give this example, forever they are going to remember this particular example. This is how we need to correlate the societal and the day-to-day activities with our teaching. Then comes the diagram which they have to draw in the journal. And yes, coming to the last part of the biosystematics and biodiversity, if we don't protect our planet, this is waiting for us. Why we are involved in teaching the biosystematics and biodiversity? We must know what is around us. We very unfortunate to say over here that nowadays apartment concept or the we can say flat system is there. Even we don't know who is the other flat. We don't know how many people are there in our apartment. But as far as the biosystematics and biodiversity is concerned, keep the doors open. And if you keep the doors open, you will be able to keep the minds open. And if you are able to keep the minds open, you can grab the subject very nicely. So with this, I come to the end of my presentation and I request the host to take up the dais. Thank you, everybody. I request Dadav sir or yeah, please go ahead, sir.
Thank you, sir. Thank you. So topic for open for the discussion. Any doubts or queries to any faculty to ask to our today's resource person?
All our faculty members, with due respect to all, I don't think so there should be any doubt, but this is how my way of taking the subject to the student is reflected over here. Yeah, thank you very much, sir.
Okay, sir. It's immense pleasure to propose vote of thanks. So, first of all, I am thankful to the today's resource person, Professor Dr. M. P. Bilaway, sir, Department of Zoology, Shivaji University, Kolhapur, for delivered to descriptive talk on the biosystematic and biodiversity. And the practical course. And thank you, sir, for giving your precious time and guiding to us. I am also thankful to the all faculty members for their attendance. And I am also thankful to the head of the department and vice principal of institute, Professor Dr. V. I. Deshpande, sir, for always inspiring to us for conducting such a program. And I'm also thankful to the Honorable Principal Dr. B.T. Zadav, sir, for supporting to organize such a program. Thank you, all of you, once again. And with the permission of organizer, session first is over. Thank you.
Thank you, one and all. Thank you for joining in time. Thank you, everybody. Do take care of yourself. Bye-bye.
Okay. Thank you, sir. Second, for the session second, our resource person is the Professor Dr. V. Y. Deshpande, sir. So I request to the Dr. A.J. Salunke, ma'am, our second resource person. Over to you, ma'am.
Thank you, sir. Good afternoon, all of you. It's my great pleasure to welcome and introduce today's resource person, Vice Principal of our college, Professor Dr. V.Y. Deshpande sir. Sir has over 34 years of teaching experience and 20 of research experience. He has recognized as PhD guide of Shivaji University, Kolhapur. Till the date, 2 students completed their PhD and 1 student are pursuing PhD under his able guidance. Sir is a chairman of Board of Studies in Zoology, Shivaji University, Kolhapur. Sir is a life member of Sir is a member of IAAB, NGO, ENCA, and ROMO. Sir is a life member of Association of Zoologists India. Sir is published and presented 36 international conferences. So, with this very brief introduction, I welcome Professor Dr. V.Y. Deshpande sir. Now, I request Professor Dr. V.Y. Deshpande sir to start his talk. Thank you.
Hello. येतोय ना?
Hello.
Hello. Good morning, everybody. After the initial talk by Dr. Bilave, actually we don't come to the actual topic. So all of you are aware, other than the new yet to come, I hope. And after that, from this year, we are introducing MSc at fisheries in the MSc 1 level from 21-20. Next year, in the year 22-20, on to MSc Part 2 Fisheries, and we are going for BSc 1 Fisheries. So, in the year 22-23, along with the zoology, our department, under the guidance of Professor Boite Madam, will be running courses. One will be zoology, right from BSc 1 to PhD, and second will be fisheries from BSc 1, not grantable, as far as the syllabus is concerned. Professor Bilave has already talked something regarding L101 biodiversity and biosystematics. So those of us who are familiar with the MSc syllabus must be familiar with all these things except number 404. The remaining three papers are similar to the zoology paper, except instead of zoological examples, we'll be opting for examples from fisheries, aquatic animals, and so on. So those of us who can teach zoology will also be able to teach fisheries as far as first three papers are concerned: biodiversity and biosystematics, ecology and environmental pollution, fish cell and molecular biology. The paper for fisheries, and we require someone in fisheries or a PhD in fisheries. Biotechnology. So similar to the papers, we move away for fisheries. So the specialization that we are planning and get the syllabi accepted in the board of studies and academic council, followed by governing body. In fisheries, we are going to go for specialization in freshwater aquaculture management. So this will be a new specialization, which is the specialization which will be different from those which are present in Aurangabad, those that are present in Nagpur, the ones which are present in Mumbai, CIF, and those which that is fishery specialization in Shivaji University, Kolhapur, where the specialization is at. And what we are keeping in view is the amount of freshwater bodies. During my first talk for the FDP of all the 7 subjects, the common talk said that what this semester also fish disease management, fish quality, assurance, refrigeration and equipment, engineering, legislation, fishery marketing and finance, fisheries extension education, and the first ornamental fish production and management, freshwater fin fish and shellfish breeding and hatchery management, fish population dynamics and stock assessment, and fish nutrition and feeding technology. So all the yeah, of my these papers are concerned with nutrition, control of diseases, marketing, the rules and regulation, the quality assessment, and some innovative, I'll say, culture methods such as ornamental fish and pearl culture. The Professor Boite ma'am was saying yesterday. So that was the syllabus of BSc, and we are following it in fishery. SFT 103, fish cell and molecular biology. The remaining two got from Shivaji University, Kolhapur. So even though this paper is the same as our zoology, everyone is familiar, everyone has learned all these things themselves. All of us in the teaching field have gone through the syllabus. We have already answered the full length questions, and we have undergone the practicals in our MSc classes, and those in the Vice Institute of Science have taught this in zoology. Only difference will be this is concerned. So today I I'll go through that. So unit number 1 is membrane structure and function. We have kept the same syllabus as that of zoology, and the learning outcomes are also similar. They must the students must understand the role of membranes to protect the cell. So structure of membrane, which I'm sure they have been studying right since their 11th standard, followed by BSc 1 and so on and so forth. Then they'll be able to explain the models of membrane structure and diffusion of molecules, and they'll know the importance of cell to cell communication. So all of us are familiar, but I see this diagram. As far as the fishes are concerned, the membrane is similar to the membrane in case of higher vertebrates, uh, for that matter, other vertebrates. But there are a few adaptations, there are a few exceptions. I am only giving membrane where the temperature of water is less than 20 degrees normally, and it may go down to -40, -60, and in summer it will be 5, less than 5, just about 5. So in these fishes, there is modification in the membrane, that instead of the viscous layer, it has become fluid. Instead of saturated hydrocarbon, it has become unsaturated hydrocarbon tails in the lipid layer. And if some of, and then omega-3 lipids. So this is lipids are present, hydrocarbons are present, lipids are present, and that is why these fishes are in great demand for their omega-3 lipids. The second thing related to membrane and skin of the fishes, than other animals is the first part. Osmosis is a real problem, and a problem to both the fishes. On the left hand side, we see freshwater teleost, and on right hand side, marine water teleost. In both these fishes, both are concentration of salts present in the surrounding medium differ in both these cases. In fresh water, the amount of salts present inside the body are more, and that is why water will be continuously entering the body. So this is something different from other mammals. What we teach in zoology as far as mammals are concerned, syllabus of MSc 1. So this is not a problem in mammals, but as far as the freshwater fishes are concerned, water is continuously entering inside because of osmosis due to the difference in concentration of salts and water. The fish will be losing salt, and as you can see, the urine becomes dilute, and there is a removal of water every now and then. And on the other hand, a marine teleost, what is happening is the concentration of salts outside the body are leaving the body. So the problem is exactly opposite to that of the freshwater fish. In the freshwater fish, water has to be removed every now and then. And in the marine water fish, water is continuously being removed without doing anything. And that is why the urine is made concentrated. And if you see the green part in front of the mouth, and above that, the red and the blue arrow, The marine water fishes have to drink water. They have to drink water continuously because they are losing water to the skin. Then cell adherence, gap junction, the extracellular matrix, everything is same in all mammals. Let us not repeat all those, the students. But the last point out, the projects can be given to the students regarding the extracellular matrix, which is useful for repair of fins of fishes. Then very good work is being carried out today by the ichthyologists regarding the repair of heart transplant, and the model fish is zebrafish. So this is one aspect where some faculty can contribute, and some students may take this particular subject because in the fish there are the fishes which are available in the market. There are so many parts which are to be waste, and they will be having this ECM, and we can get it almost free of charge, and we can use it for research experiments. Then this unit number 2 is secretary part is nothing is different mammals and the fishes. So structure of ER, then transport, ribosomes, polysomes, free ribosomes, everything is similar. All the fishes also have these organelles, and then vesicles involved in intracellular transport. Again, this is true as the fishes are vertebrates. They are also having hemoglobin as a carrier pigment, and because 9 or 10 years for the zoology. And they will understand the role of ribosomes in protein synthesis. So this is all repetition from zoology. The basic of all this topic has already been considered for the students from standard 11. Then cellular respiration, the topic is same. Fishes also have peroxisomes, they also have endosomes, they have lysosomes, they have polymorphism. There are some interesting aspects regarding proteosomes. This is a very good topic on which research is being carried out. I think I have a slide in after this. Then mitochondria also, it is a universal organ present with the same function everywhere. And detoxification role of cytotoxins, say P450, everything is same, the xenobiotic transformation. Learning outcomes, students will be able to explore the structure and function of cell organelles. This is a wonderful outcome which our zoology will be able to understand in a better manner than any other student or Pune or Mumbai University in our neighborhood or at college level. Why? Because I come to the slide very soon. Then the role of cellular activities detoxification occurring in the body. And the third point is related to detoxification, where many of the PhD work is being carried out in our department regarding the toxic effects of some chemicals, some So this is where the topic regarding degradation of uric acid in fish liver peroxisomes. So peroxisomes, this is the current research trend. So we can concentrate, even future students can be given a few projects related to this particular aspect, where peroxisomes of marine fish liver can be given, and research. Dr. Karanskar has, I think, carried out practical related to this lysosomal activity acid and alkaline phosphatases and their role in detoxification. And I think even the current PhD students are having a few topics related to these lysosomes. Now, this is where the application comes in. If we are going to produce fish farm managers, and they are going to be experts in maintaining a fish farm, then different aspects that I would fish farm management, regulating protein degradation. So as far as the protein production is concerned, so research articles are present. The process is well studied. We teach the students how the mRNA is formed, and then how the tRNA is and whatnot, and then the protein is synthesized as per the requirement, structural proteins and functional proteins, and so on and so forth. But the degradation of protein part is not yet properly understood, and this is what the fisheries farm, the specialization that we are opting for, are studying. And the last paragraph, I've put it over there, that the mRNA levels in LSD, that is low stocking density group. LSD means stocking density. Low stocking density groups were significantly higher than those of high stocking density groups. These results illustrate that fish cultured in lower stocking density would exhibit a greater growth rate and fast protein turnover in muscle. So, the aim of producing freshwater aquabiologists at fish farm managers is to increase the production of fishes, lakes and streams and rivers and freshwater bodies present. So many farm ponds, government of India and locally subsidized by Jilla Parishad. So this is important. What is happening is, if you more number of fishes, the production may be more. So it's a somewhat anomalous relationship, because less number of fishes put in a tank will give you more production and will give you more profit. So these topics in the syllabi must be correlated to the application of these particular topics for the betterment of fish production. Then last, then cytoskeleton and so on and so forth, signal, and the students will be able to get clear idea about electron microscopic structure of nucleus, various components. Then they will be able to explain the pattern and packaging of chromosomes in nucleus, and then know the and its role during cancer and cellular signal. What I was saying a few minutes ago related to why our students are having an advantage over other students in the area or in the university is as far as the practicals are concerned. So practicals are similar to logic and demonstration of cellular material, that is collagen and elastin. So I've given a few diagrams and methods which can be in the laboratory in our department. There are a few chemicals which are required, but as we are going for fisheries at MSc, I'm sure we will have to procure these chemicals. And this is how very beautiful slides can be prepared where all the different colors, if you see 4 or 5 colors are present, and they will show nuclei, elastic fibers in black color, in red color, collagen and reticular fibers yellow color, and mucins in blue or green color. So this is another method, one step trichrome, that is Gomory's method, and these particular fibers, collagen, they are used for extraction using speed and demonstration by the students. Then this is a new name for our practical which we have been carried out since our BC3, and this is valid also for fishes. But in addition, there are a few the underlined blue color part, that is mucopolysaccharides from skin of fish Libio roita. They can be extracted, not only demonstrated. We have some, till now maybe BSc 3 or MSc. We are showing the students. We are going for histochemistry, histopathology, and moving to histochemistry, where we demonstrate the presence of these substances using different stains at different pH, 1 and 2.5, and so on and so forth, using Fulton pH and so on and so forth. So this was only demonstrative. But if we go for the we can extract these, and these will form very wonderful and interesting practicals. And this is what I organelles. We have an advantage because all of us know our institute has established and bought and established a scanning electron microscope. Our campus just cabin in front of the canteen in the garden. I hope all of you have at least visited the place once. It is a totally air-conditioned room. Dr. Mujawar from physics department is looking after that instrument, and we are carrying out outsourcing where graphs are available. I think the rate is very minimal for students from our institute, and we charge less than 50% of what is charged from the others. And you are able to see a cell. So I have dropped the diagram. So this is how we will microscope. If you provide a slide to Mujawar sir, he will give you a photograph showing the nucleus, the nucleopore, the Golgi complex, and mitochondria and different cell organelles. So instead of simply showing the students the organelles in a slide, if we can just take them scanning electron microscopy, we are also able to visit and see all these organelles in our laboratory. Then effect of tonicity solution on plasma. So this is an application. So the topic related to the previous one, the fragility test of RBC, and osmotic resistance, and so on. So what we actually do is we teach the students their cell wall will be destroyed at different temperatures and different concentrations. But as far as the fisheries is concerned, this particular topic will be important because the research going on right now uh, in freshwater as well as marine water aquaculture. I'll be back in 1 minute. Uh, So, sorry for the interruption. So, I was saying that instead of simply teaching the students the fragility test of RBC and osmotic resistance, what the scientists are doing are the application of this particular practical. They're studying individual plasma components, stabilizers found from glucose, proteins, and ascorbic acid. And what happens is this, if the temperature is maintained, if some chemicals are used which are not harmful, the longevity of the fish flesh is increased. That is, we can keep the fish without it getting deteriorated, without losing its nutritional value for a longer duration. So this is a application of the practical. So thank you everybody. Books, as far as the books are concerned, the list of books given in our syllabi is mostly related to zoology, but I have prepared a new list related to fisheries, and I'll be putting it up on the website very soon. So at we come to the end of this particular paper and what is to be done. I think practice. Benjapati madam, South madam, Kada madam, they have handled the practical and the theory portion, and many of us have contributed by taking a few lectures. So this is what we are supposed to do for MSc One Fisheries. Specialized papers for fisheries. Remaining will be similar to those of zoology. Only thing is the applications will be different, the examples will be different, and speaker from Ratnagiri and one from Kolhapur. They will be dealing with paper number 2 and 4. So thank you for your patient listening. And we can discuss something if any one of you wants to say something. So I'd like to on the 7th we are going to celebrate Teachers Day. Now, and on that day, we are publishing our e-books, which are based on syllabi of different courses. And in due course of time, the fisheries textbooks in the form of e-notes and e-books will be coming out in say a time period of 1 month or something. So even if reference books are there, I'm sure the first few batches of teachers, not the students, I'll say teachers, will be benefiting. I will announce or request the interested faculty to come forward so that we can write those books. We have completed 2 or 3 of them, which will be publishing on Teachers' Day. Yes, Teachers' Day.
Thank you, sir. Uh, oh, I'm thankful to the resource person for this Professor Dr. V.Y. Deshpande, sir, valuable information on the fishery syllabus. Thank you, sir, for giving your precious time and guide to us. And I'm thankful to all the faculty members for patient listening. And thank you, one and all. And with the permission of organizer, I declare that the today's sessions and faculty development program of MSc First Fisheries carry on the Monday, 6th of April from 11th a.m. So we meet on the